again, the question is not if it's deuterium depleted. Yes, we know that it's deuterium depleted. It's just how you control over time and electrolyte or electrophoresis, electrode contamination and isotope effects, and how stable it is when it comes to clinical trials. And how a compounding pharmacy, which every hospital, because you cannot just go to a hospital and give patients deuterium depleted water. It has to be in a form of an IV, or it has to be in form of a therapeutic approach, maybe oral administration, but a pharmacy has to assemble, they have to produce those IVs or whatever. At least that's the law in the US, that the university or the clinic that runs clinical trials, they have to have a pharmacy that assembles and label them with expiration dates and so on, the product itself. And this is, I think, where the technology may become somewhat, maybe in the UK, you can get it approved by IRB protocols or procedures using a combination of fractionation and flame ionization, but practically that's something you need to clarify before you get to the clinical trials, like how, because Gabor's water, the fragmentation, the physical process, since you fragment and you distill water, there's no potential contamination, simply because this is one of the most efficient water purification method, is practically to generate fractions of water vapor, and then condense that water. And those usually easy to submit as part of clinical trials. Neither the Clarivia type, meaning that it is by a wastewater of a atomic power plant, or the process of making heavy water, the side product, or any other method, those are a little bit harder to push through IRB protocols, procedures, simply because those are a little bit hard to control because of the many components that are involved in a distillation tower. You have to look for two things. One is the vacuum, the second one is the electrolyte plates, or the condensation plates, where they are, and you just measure deuterium content in the water. If you have to electrolyze, and there are, again, as you know, there are, based on the water purity and based on the additional conditions, variations in electricity or whatever other components or factors that are involved in the process, you may have to explain those to the IRB guys, the institutional review board guys. But again, I'm not involved in the technology part, so I'm not sure exactly how to address those issues. But it's just clinically their efficacy is somewhat, again, questionable, because your body produces, your gut bacteria and yeast produce hydrogen gas as well. And the reason why they produce hydrogen gas is that because of and I'm sure you have you seen my talks?
Yeah, some of them, yeah.
Yeah, so bacteria and yeast, for their growth, they produce hydrogen, or they expel hydrogen from the cytoplasma through these ATP hydrolase dependent proton pumps to retain deuterium for their constant growth, because cell growth depends on deuterium content. So these yeast and bacteria, they retain deuterium while they are pumping out hydrogen or protons. And this hydrogen gas that is produced in the gut as part of the digestion process, this is why it's hard to dose hydrogen gas, simply because it reacts with oxygen very rapidly if oxygen is activated. And red blood cells carry activated oxygen endothelial cells, epithelial cells have electron transport chain that activate oxygen, and any hydrogen gas that is absorbed through the gut or some other tissues, they may end up forming metabolic water right in the endothelial or epithelial cell layers. And it seems that it's somewhat easier to carry deuterium depleted water or hydrogen through food, that would reach a body or host cell mitochondria based on substrate that are absorbed and carried in various forms. So hydrogen gas does not last too long. Maybe after the first passage through the epithelial cell layer, it's already forming metabolic water. It may protect very well the surface lining cells and cell layers, but we don't know how far it penetrates into tissues, for example, liver or muscle tissues, after passing through so many cell types and membranes and so on. And that's the question that we don't know for sure. But obviously, we can set up a MRI-based diagnostic procedure. If you guys are involved in proton scanning, we can actually measure those deuterium content in those tissues based on MRI images, because we have a program for that. So we can actually take this further just to answer these questions. So there are a number of important points that we need to consider or we need to contemplate of how to, interpret some of the findings that you see in hydrogen gas or hydroxy gas treated patients. And it's interesting because hydroxy gas is produced in your mitochondria as well. So again, is it coming from food or is it coming from the gas that you're applying? How fast or how deep it penetrates into tissues, and how easily you can access target tissues, and what are the common deuterium depleting effects of these remedies or these interventions that actually deplete deuterium for the system, for the host, but it may not reach the target tissues simply because of the chemical, inorganic physics and chemical characteristics of these gases, practically. So in my opinion, the safest way and the most efficient way to reach every mitochondria in host cells and in target cells is practically through food, low deuterium food, which is a natural ketogenic diet. If this fat source is natural, of a natural source. And again, mitochondria produce hydroxy gas simply because of this stabilization. Have you heard of the I gave a talk in October about the stabilization, proton destabilization in mitochondria. And when it happens is you actually produce hydroxy gas in your mitochondria, and you make the proton to disappear in so-called quantum vacuum, which is the proton stabilization process that is how mitochondria recycle water. So the question, because of these nano confinements in mitochondria, those crypts, and if you haven't heard that talk yet, it was last October in Budapest. You may want to listen to it because that gives you a number of new information of how mitochondria process water from hydrogen, or water made from hydrogen introduced in the form of gas, and also how your mitochondria produce hydroxy gas by water's proton destabilization, that means you may have to go into a little bit deeper as far as its biochemistry arguments go, simply because there are some other topics or other physiology or biochemistry that you may need to review before you start a clinical trial.
Yeah, one question I wanted to ask you about ATP is what would happen if the ATP would take deuterium rather than hydrogen in? Would it break the engine of the ATP, or what would happen?
Are you talking about ATP or ATP synthase?
Oh, ATP synthase. So it's kind of what would happen if it's taking like the double-sized deuterium in instead of the hydrogen? Would it break?
Well, it can't take it. Because it breaks down these. They start stuttering. Stuttering is practically when they start wobbling the engines, because you imbalance. You just practically, there are 12 protons in the mitochondrial ATP synthase. They spin about 10,000 rotations per minute. So it's a very high velocity nanomotor. And if you somehow hit it with a deuterium, it would actually, because of the disbalancing effect of deuterium, the double weight is just going to rip the nanomotor into pieces, meaning that it's just going to break the nanomotors. So we believe that glycolysis and all the biochemical reactions that are part of like our biochemistry is, first of all, those reactions are there to deplete deuterium in substrates, in sugars and proteins and fatty acids, and also their purpose is to protect the mitochondria and the ATP synthase nanomotors from deuterium. And even though if deuterium makes it in the mitochondria, the nanomotor has its own gating mechanisms to collect heavy hydrogen isotopes like deuterium and tritium. And it's called the oligomycin sensitive protein part of the nanomotor itself. And we believe that this is how oligomycin works. It actually deprives these bacteria's ability to retain deuterium using these nanomotors, and they actually die. So the nanomotors in nature are designed to filter out deuterium from hydrogen, and it can go both ways. In bacteria and yeast, which are constantly growing, they use these nanomotors to actually pump protons outside of the cell while they are retaining deuterium. And there's a paper back from the 1990s that describes this process, and they talk about deuterium is not able to replace protons in ATPase proton pumping procedure. So this nanomotor cannot take deuterium, and that's the same in mitochondria. So there are serious gating mechanisms that are involved in the process. Hydroxy gas is produced in mitochondria in these water nanoconfinements, hydrophobic nanoconfinements, which are membrane-based nanoconfinements. I delivered a talk on this, quite extensive talk in last October, which you may want to review before you position either hydroxy gas or hydrogen gas in your deuterium depleting scenarios, because the biochemistry is a little bit more complex of these procedures.
This is great, thank you. I mean, one question I wanted to ask is about to do with childhood and pregnancy with regard to deuterium. What role does it play in childhood and pregnancy? And are there contraindications or any relative contraindications that we know about with regard to this?
Well, again, our ancestors and our ancestry is in a deuterium depleted environment, meaning that the caveman was eating fat mostly in bone marrow, and the pregnant mom and the babies, they have a different deuterium depleting scenario. So you can give them deuterium depleted down to like 125 ppm or 120 ppm, which is like good rainwater if you live especially at high altitudes. And the baby and the mom, they have a specific way of depleting deuterium, means that they don't deplete as much from the available deuterium that is in your body fluids. And as a result of that, the placenta and the constantly dividing fetal cells, they have enough deuterium to drive the cell division processes. And that's the same when the baby is growing and drinking milk and milk sugar and loaded with deuterium. Those are again to make the baby grow, but they have it can be a deuterium depleted environment because they have their own deuterium processing, collecting, discriminating, filtering mechanisms, so they can retain more deuterium in the growth phase. And these are again biochemistry related mechanisms. It seems that we have our biological biochemical abilities to retain or get rid of deuterium. The only problem is that when you cannot really deplete deuterium sufficiently, your deuterium depleting potential or your food or your water is too loaded with deuterium, and your capacities to deplete deuterium are not able to deplete deuterium sufficiently. So you suffer significant mitochondrial damage. And we know that happens in cancer, in obesity, in diabetes, in degenerative diseases. Those are all part of this metabolic crowding process or molecular crowding process, where mitochondria are broken then because of high deuterium in food, processed food, and farm-raised animals and grain-fed animals, and drinking 2-3 liters of water, which is senseless, high deuterium water. That's usually the doctor's recommendation, which is bullshit. You actually deprive antidiuretic hormone synthesis. ADH synthesis is important for regulating deuterium disposal through urine, and it also affects your ATP synthase nanomotor mitochondrial functions. This is the oxytocin or vasopressin kind of mechanisms. Those hormones would be very important, but usually because doctors make patients drink unnecessary water, with high deuterium, their ADH levels are low, and they are practically operating in a diabetes insipidus kind of state. Most of the population, because of the excessive water drinking, is just, they have so many comorbidities now. I think the largest, most crucial comorbidity is high deuterium with different tissue and disease manifestations, but they are all based on tissue-specific deuterium accumulation processes. So those have to be addressed in these clinical trials, and find out what are the most efficient approaches to actually deplete deuterium in those patients. It's through food, it's through hydrogen gas, or it's through hydroxy gas. Practically, these are the important questions to ask in the first place. And then you can approach the medical problem with the appropriate deuterium depleting biochemical scenario.
Great, thank you. That leads me nicely to the question I was going to ask you next, because we're looking at embarking on research into the molecular hydrogen deuterium depletion. So one of my questions was, what kind of research would you like to see done in this area? What would you think would be the most beneficial?
I would use an MRI method. It's a low. I already gave a talk about this. Again, I can send you these talks so you can specifically review those talks for your clinical trials. But I would use deuterium depleted water, low deuterium food, ketogenic diets, and hydrogen gas or hydroxy gas, and I would see how MRI images based on a standard procedure on a low magnetic, it's usually a T1 sequence that we run in low magnetic fields 1.5 Tesla, because that's practically a deuterium measuring device, because it's based on proton spin. And I published and I talked, I gave talks about these. But if you could combine the two methodologies, we could develop a common platform how to address these questions. First of all, how deep hydrogen gas or deuterium depleted hydrogen gas penetrates into host tissues. It may have a very good, very desirable clinical effect just to prevent gut leakage syndrome, because I'm sure hydrogen gas in the gut will be beneficial for epithelial cells and surface layers, which is a huge accomplishment in some of these chronic disease scenarios, to actually maintain the mucosa and the gut flora, the microbiome, in a good shape. And then you're going to see a lot of response as far as chronic diseases, joint pain, just like what you listed before, what you told me, like you saw very clear effect or clinical benefits in these particular arenas, but disease states. But they can all be explained by various effects of hydrogen, deuterium depleted hydrogen gas, hydroxy gas, and deuterium depleted water and deuterium depleted food. And we can actually interpret this in the clinical scenario, simply just going after these MRI images and see what density, what proton densities are changing in whole body scans, and how deuterium depleted water or hydrogen gas is distributed. Those are, I would say, the most efficient ways of looking at this in your translational clinical studies that you are trying to accomplish.
Yeah, and what do you think? What do you think will be happening in the future for this for deuterium depleted water? I mean, what do you see happening? Where do you think it will go?
Well, right now, you probably know that there are now papers out, clinical papers, that actually address in the discussions. They see clinical effects, and it's always better explained by deuterium depletion, but we just have to find how those are, fitted with the clinical scenarios that they see. And there are, like, I would say 5 or 6 papers now in the medical literature, clinical papers, that actually show that the intervention or the therapeutic approach that they used is actually depleting deuterium in those patients. And those can be used as the mechanism or explaining the beneficial effect of any kind of treatment. And again, transformation, cell transformation is very much dependent on deuterium loading. And so you can see those clinical papers coming out. I'm editing right now 2 major papers that will discuss deuteronomics. There are 5 or 6 papers already out, which can be used for your references, and you can read those and you can read the discussions. I'm going to send you, because you sent me your email, so I can send you those documents. And it's going to be the mainstream medical, I would say, biochemical medicine or deuteronomics. I'm just finishing a textbook on medical deuteronomics, which will, list and describe these deuterium processing biochemical pathways that to understand disease better, and to use these as clinical remedies or clinical interventions. But again, many call this mainly in the integrative alternative medicine field, as we probably this century, this decade is going to be devoted to a lot of hydrogen, proton, deuterium, tritium type of research, at least, clinically interpret these. And deuterium depleted water will be, with some other considerations, deuterium depleted food through metabolic water production will be an important part of medicine. It's just practically how we can actually push this through the regulatory agencies, because pharmaceutical companies don't like when you offer a better treatment, to replace their drugs, and how they are. So I probably don't have to explain this to you, but it is visible in the medical literature. It's if you just type in, if you have your computer, if you just type in depletion and nature, it will give you some clinical papers already published in Scientific Reports, which is a Nature publication published by Nature Group. And there are some other important papers that I'm going to send you. And if you look at those discussions, those are very clear and very specific of how that particular disease process was addressed by deuterium depletion based on the treatment that they applied, which they couldn't explain otherwise. And those are very important, I would say, clinical papers in the medical literature that you can use for your own research or design your own studies. But I think this is going to be applied in many areas of medicine once we have the clinical applications or clinical recommendations or the clinical studies that need to be done before this can be proven in controlled double-blind studies that this is actually a good therapeutic approach. And we can also look at the various mechanisms and the various approaches to deplete deuterium. Some may be hydrogen, hydroxy gas, some may be water-based, and some may be food-based, based on the disease conditions that we are working on. But I think this field is going to be big in the next, I would say, 5 to 10 years.
Mm-hmm. Yeah, we've certainly found that, for us to do a clinical trial on unhealthy subjects with for deuterium depletion requires 8 months of planning, NHS ethics approval, and many millions of pounds of funding to do that kind of research. So we're focused on just at the moment to do unhealthy subjects to study the inflammation just with the hydroxy gas, because we can supply a machine to the university and they can use it many times. But we think that it could be a good way to make DDW in the future, possibly, but we have to test, I was going to ask you some questions about COVID-19. I don't know if you're sick about talking about this now, but
sure.
Because one of the questions from Dr. Maria in our team was specifically to explain why is it that in COVID-19 cases there is insufficient oxygen delivery to the tissues, and what she's asking is, from a clinical point of view, this is not lung function related, but red blood cell insufficiency to carry oxygen, less oxygen to the tissues and less metabolic water. So is that understanding less metabolic water and less energy production in the cell, and therefore cell death and organ failure? Is that correct understanding, or are we missing?
Yeah, so have you seen my, with Dr. Petra Dorsman, have you seen the video that we put together for the American clinicians? If you go to a YouTube channel, if you go to YouTube and you type in deuteronomics, DEU, deuteronomics, there is a channel and it has a video which explains most of what you asked me.
Yeah, we're on the right lines of understanding this kind of. Okay, I will do that. I'll put the links in this video as well.